Hello, my name is Colin, and I'm going to be sharing with you some research that has used genetic sequencing to look at soil antibiotic resistance in a site that's experienced a recent wildfire. This work is being conducted out of Brent Ewer's lab in the botany department under the mentorship of Mario Bretfeld and Lena Guadagno. So here's the site named Chimney Park in southeastern Wyoming. This site has been subject of ecological research since 2009. And so using eddy covariance, EC here, and vegetation plots, researchers have been looking at the bark beetle epidemic. And then in 2018, the Badger Creek fire came through and left this mosaic of different burn severities. And that prompted an overarching project to look at the dual disturbance of bark beetles and wildfire. Now, the research I'll be presenting you focuses specifically on the effect of wildfire. So the different sites in here, there's a stand replacing fire, and in here there is complete tree mortality, and this is the most severe burn in the area. It's surrounded by understory sites, which is a less severe burn that went through the understory but left the canopy mostly intact. And then adjacent to the burn sites is a no fire site, and that's really useful for making comparisons between the different sites to have that no fire for reference. So here are some hypothesized effects of the fire. And for our purposes, what's most important to note here is that both the understory and the stand replacing site are going to experience similar impacts from the fire. But the stand replacing fire being more severe will impose more significant impacts. And the no fire site is hypothesized to remain in a mostly steady state, and that of course will make for a useful reference. All right, so why study soil microbes, antibiotics, and resistance in light of fire? Well, in terms of antibiotics and resistance, it turns out a lot of the key compounds and organisms and resistance genes that are found in the clinical setting are also found within the soil. So in terms of fungi, we have penicillium, which were discovered to produce penicillin. Also are aspergillus, which produce a variety of different antibiotic compounds. In terms of bacteria, we find actually the most prolific antibiotic producing organisms in the actinobacteria, which naturally produce 2/3 of all the known antibiotics. You can see here on the right, they form these multicellular filaments, and to reproduce, these cells are pinched off to form free spores. Also within the bacteria are the bacilli and clostridia, and these are two closely related groups, very closely related. The bacilli produce a variety of different antibiotics, and Clostridia do not, but both groups form very similar endospores. Now you can see on the right here, Bacillus subtilis in different stages of sporulation. So here's the vegetative cell, and then the endospore is formed within the mother cell. And so there's a great deal of condensation of proteins and membrane layers that surround this spore and give it a great deal of durability. And so that's going to be really important in relation to fire, right? Because that durability has been demonstrated to confer heat resistance. And the spores of actinobacteria may also be able to confer some level of heat resistance. All right, so on the left here we have the life cycle of actinobacteria. And it's important to note that antibiotic production and sporulation are part of the normal life cycle. This is also the case for bacilli undergoing sporulation, where antibiotic production precedes the formation of spores. But there's some key differences between the two different sporulation programs. And the most crucial difference is the end product when it comes to resisting heat. So you can see here on the right is an endospore characteristic of bacilli and clostridia. You can see it has a number of these protective layers formed by protein and membrane. Now, the free spores that are formed by actinobacteria are going to have less protective layers. And so that has led us to hypothesize that those endospores produced by bacilli and clostridia are the most likely to survive the heat of a fire out of really any cell type that's out there. And the actinobacteria spores may have a competitive advantage over some different cell types that do not form a spore. All right, so these questions are being approached using metagenomic sequencing. So we have taken soil samples from each of the 3 sites, standard placing fire, understory fire, and no fire, and they've been shipped off for sequencing. And as part of that process, soil is extracted, DNA is extracted directly from the soil and then sequenced. And then using a great deal of computation, these short sequences from the environment can be assembled into portions of genomes. And using reference to databases of known genes and known genomes, there can be identification of phylogenetic groups and also functional genes such as antibiotic resistance genes. And then by comparing the relative number of reads of different functional genes and different genes related to phylogenetic groups, we can then compare the relative abundance of those groups and those genes. So here are some results from the 2019 sequencing. And it's important to acknowledge that there's a low number of replication, but this sequencing yields an enormous amount of reads. And we can also fit these sequences into the broader framework of the data from Chimney Park. And I'll talk about that a little bit later on. But as you can see, there's an enormous amount of diversity. And this is just looking at bacteria and archaea. So when we look at those groups of interest, there's not an especially clear trend with the actinobacteria across burned sites and the no-fire site. And that sort of fits with them forming a less heat-resistant spore. But when we look at bacilli, it looks like clearly they were enriched in the understory fire in that moderate heat, and they might have done a little bit better than competitors in the stand replacing fire. Now looking at Clostridia, which also form very similar endospores, it also looks like there was a slight enrichment in the burn sites compared to the no fire. Now looking at fungi, and specifically those 2 antibiotic producing groups, the Penicillium and Aspergillus, we see that in 2019, these 2 groups actually made up a small fraction of the total fungal population. This is relative abundance in terms of the total fungal reads. But in 2020, these 2 groups made up to as much as 100% of the reads from a given site. And so that's pretty curious, and that's definitely something that we'll be digging into as we analyze this new 2020 data. So how does all this community composition line up with antibiotic resistance? Well, in 2019, there were 130 different antibiotic resistance genes identified in the understory fire, but none were identified in either no fire or severe fire, standard placing fire. In 2020, there was a much more even distribution of resistance genes identified. And this was more even in terms of the different types of resistance genes, which is being depicted on this axis, but also in terms of the number of reads, which indicates the abundance of resistance genes. And so that will be something that will need to be analyzed in more depth going forward. Okay, so the big take-home points here are that soil microbial communities are reservoirs for resistance, and that arises because many members naturally produce antibiotics. And when there's antibiotics present, resistance will certainly be present as well. There's an interesting physiological connection between sporulation and antibiotic production. They're often part of an interconnected regulatory network, and that sporulation could confer an advantage for surviving the heat of a fire. And so that's what connects the antibiotics to the sporulation. Now, looking to the future, as we look from this site, the Chimney Park site, into the Mullen fire that occurred this year, in the future, we'll be doing more in-depth analysis of the sequence data and comparing 2019 to 2020, but also fitting the sequence data into the broader framework of what we know about Chimney Park. And that's based on the eddy covariance data and a variety of soil moisture, temperature, carbon, and nitrogen measurements, and trying to pull out relationships there. So thank you for your attention, and I look forward to discussing this with you further.